Hey guys, today I'm going to be talking about something I've been experimenting with a little bit in FMRI called k-means clustering. Now, you probably may have come across this at some point in statistics courses. It's not a terribly complicated concept. It's just trying to basically determine whether a data point belongs in one cluster or another cluster, kind of based mainly on how close they are to certain centroids or centers of mass for each of those clusters. Now, this is kind of a rough draft of how I'm applying this right now to a study I'm doing. I don't know if I'm exactly doing it right. Some of the things I'm doing is a little iffy, but just kind of walk you through it. I'll walk you through my reasoning and see whether it gives you any good ideas of your own to apply to your studies. Now, talking just really quickly about what k-means clustering is, and this is from the k-means documentation on MATLAB. Let's say you just have, say, two dimensions. Okay, this is two-dimensional data. There's a y-axis, there is an x-axis, and let's just say we have a ton of data points, so xy data pairs. If you just eyeball this, you can make the case that there might be two distinct clusters, one down here, one up here. And that's all the k-means algorithm does, is basically says, okay, if there's a centroid or a center of mass for this cluster over here and one over here, to partition this into two clusters, how can I best assign which data point belongs to which cluster? Okay, so which data point, is it more in the red group or in the blue group? Which one is it? Okay, you could not just have two clusters, you could try to partition it into multiple clusters, and you could also be dealing with multiple dimensions. So imagine there's another axis coming straight out of the screen towards you, so it'd be three-dimensional data, and you'd have little clouds of data, and then you'd be trying to clusterize based on that. All right, so now applied to this fMRI stuff, and this is going to be pretty crazy, but see if you can follow along with me as we go on our journey together. Always exciting. Now, there are a few things that we need. So we need XYZ coordinates for every voxel. In this case, I'm using a whole brain analysis. The same thing would apply if you're using a restricted region of interest. You would just be getting the voxels for just that region and nothing else. And we also need, say, all the regressors of interest. We need to put them into a single matrix and then use k-means to determine whether each voxel belongs to one cluster or another. Just to show you kind of what I'm dealing with right now. It's always kind of hard to see everything. First thing, when I'm loading this up, I'm setting the p-value to 1. I'm not clustering at all, just so I get every single voxel activated so I can get each coordinate. Now, you may have seen me do this in a couple of other tutorials, but so what I'm dealing with four regressors, and what I noticed just looking at this is that three of the regressors seem to be very active in this more dorsal aspect of the medial prefrontal cortex, and the last regressor was more active in this region down here, more ventral anterior cingulate. And the patterns were basically reversed. If you go up here, then that single regressor isn't very active, but the other three are. Go down here, the other regressor is active, but the other three are not. Now, this is a special case where I can't use a simple interaction test because some of these regressors tend to overlap a little bit and it's not fully teased apart like that. So this was a case where k-means would be useful, and instead of doing a bunch of different interaction tests, just doing a single k-means to kind of simplify it and make it much more readable when it goes for publication. Alright, so the first thing I'm going to do here, this is going to be a two-part tutorial, and we're near the end of the first part, just giving you a background and showing you how to set it up for the actual k-means analysis. So what I'm going to do is I'm just going to click current cluster, get this thing activated, and if you recall, there's this xspm.xyz structure which will give you all of the coordinates across every voxel that's activated. So everything in the entire mask that I have active I want to look at, right? Okay, so what I want to do is I want to just make this, say, a variable called xyz. Let's just say for the sake of argument. And I also am going to, I'm also going to run the k-means analysis on a bunch of the regressors. Okay, I'm going to be looking at all the regressors and have a value for each regressor in each voxel. So if you remember this thing, there's spm_getData, and within each of these second level analyses, right? It creates, and you create, say, like one contrast, for example, and you split it up like that, you will have the ability to extract data from each individual voxel for that particular contrast. So in this folder, there's only one contrast. I'm looking at, say, the mean effect of pain, because this was a study where I shocked people because basically I'm an asshole. That's kind of what it boils down to. I tell people it's for science, but that's not true. I'm gonna shock people, yeah. And we're also going to use these SPM X, Y, Z. Okay. So this gives me a vector of all of the values for every voxel, okay, for that particular contrast. And now what I would do is do it for every other second level contrast and then just concatenate them together. So I might get a variable called Y, and I'll give it, let's say there's pain, incongruency, effects, a pain prediction error, and stroke prediction error, and to concatenate all those guys together to get this 4-column matrix right here. Okay, and the amount of rows, again, each row is a single voxel. So in the next tutorial, we'll be talking more about running the actual k-means analysis and then putting that into an image.